Good morning. This is Daryl Brugink, Executive Editor with No-Till Farmer, and I'd like to welcome you to today's webinar where we will be looking closely at how to manage your no-till residue. And more specifically, we're going to look at what you can do this fall to assist the biological system with breaking down particularly that corn residue, but perhaps even wheat residue, basically so that you've got less of that residue to plant into the next spring. Now, obviously, no-tillers want the benefits of residue. There's a lot there with nutrients and the cover it provides, but there are also some issues with residue that can make it difficult to get crop establishment into no-till the following year. Today's webinar is sponsored by Midwest Biotech, and they're the distributors of Chandler crop products for biological farming. And we want to thank Midwest Biotech for sponsoring the webinar. And today's presenter is Doug Miller. He's the Vice President for Midwest Biotech, and he's joining us right here in our No-Till Farmer offices. So it's good to have Doug here in person. I also want to thank our producer, Cole Vandermouse, for heading things up today. So with that said, thanks again for joining us this morning. I want to turn over the controls to Doug Miller. And good morning to you, Doug, and thanks for joining us here in our Milwaukee offices.
Thanks, Daryl. Good morning to you and to all the folks listening in. We look forward for every opportunity we get to work with the folks at No-Till Farmer, and especially this opportunity to follow up on our January webinar that we had, and we had a really good response. Talked about some of the biological products that are used in farming now, and this one we'd like to focus more or less on residue decay. It's an issue that we hear a lot about, increasingly so because of the amount of residue that's produced, especially in corn production. And so we want to take a little time today. As far as our background and where we're coming from, I'll refer to that January webinar that's posted not only at our website, but No-Till Farmer. You can find it on YouTube. But we'll jump in today and talk just about that residue decay process and what's involved, what are we trying to break down, which particular microbes are involved, what factors influence their activity and the rate that you can break down crop residue, not only corn stalks, but wheat stubble, other sorts of residue you might be dealing with. After that, we want to talk about how you can measure the amount of residue that's in the field, get some ballpark numbers on the amount you're dealing with, and more importantly, how much nutrient is actually in that residue that can be broken down and released for use by the next crop. And then we'll want to answer some key questions that we get all the time from farmers that are dealing with residue decay issues. Should I add sugar to help with residue decay? Does that help? How much should I use? What's going to happen if I use some sugar product? Some people are using table sugar, molasses, other sorts of things. Should I add additional nitrogen? And then, of course, we want to talk a little bit about our product, Chandler BioCat 1000. It's a unique product in the market. It's one of the few enzyme-based decay promoters, but we'll leave the sales pitch to the end and spend most of our time today talking about the components of residue decay and how to make it work better, which factors influence it, and sort of the technical stuff. And we'll leave the sales pitch to the end. We do want to use about 45 or 50 minutes of your time this morning to talk about those issues, and then leave at least 10 minutes at the end for questions from the folks sitting in and listening in on the webinar. So that's our plan, and so we'll get started. Three main components of plant residue that we're going to worry about. The biggest one in terms of volume is typically cellulose. They tend to be relatively large molecules. They're unbranched, but that's where most of the energy comes from that you're getting out of feed value and in silage, other residues that might, or plant matter that might be fed. That's also the source of the energy for ethanol production. And it's one of the biggest components that you're breaking down if you're decaying residue in the field. Breaking down cellulose is not terribly hard, and that's not usually the issue. Cellulose has more or less a cousin called hemicellulose. The chemical nature is a little bit different. They tend to be shorter molecules, but they're branched. And the hemicellulose provides a network that holds together the cellulose and the other components within the cell walls. So those two components are where you get most of the energy in ethanol production, a lot of the feed value, and it's also going to be most of the source of the residue that you're breaking down in a decay process in the field. The toughest part is the lignin. That's the third key component. Lignin is a very complex polymer. It's a very ugly, long, complicated, dense chain of atoms strung together to form these molecules. And because of its density, it's very difficult to break down. Its value is it provides a great deal of strength to the plant. And so the toughness of a stalk and ability to stand up over the season, especially after you set ears on corn or heads on wheat, comes from the lignin. The cellulose and hemicellulose tend to set in the plant earlier in the vegetative growth, and the plants accordingly are more flexible and pliable and can grow. Later in the season, it starts to form the lignin in the open spaces in the cell walls, and you can think about it sort of as the rebar in concrete. It provides the strength and stability. That also means that it's very, very difficult to break down. In an ethanol plant, the lignin represents material that simply is not going to yield very much alcohol. It's more or less used to fuel boilers and generate steam, but if we could break down the lignin part, you'd get a higher fuel yield from an ethanol plant. In livestock feeding, it's not digestible in ruminant animals, so it represents more or less a loss in feed value if we could find ways to break down the lignin. A lot of the seed companies now are producing BMR or brown midrib corn and sorghum varieties that are bred specifically to have less lignin content, so you have more digestible content in forages. And so by getting rid of some of that lignin, you improve the feed value. And in decay, the lignin often represents the toughest part of corn stalks or other residues, and often gets the blame for things like punctured tires and plugging up planting equipment because it makes the stalks tough and hard to break down. And we'll talk a little bit more about that later, but these are the three main components. In terms of their percentages in different types of residue, the cellulose and hemicellulose are the big pieces. They represent the largest shares. There are other compounds. There's some proteins, there's ash, there's some other biochemicals. There are other cousins to cellulose, xylan, and other things. They're not really important for residue breakdown purposes. So these percentages don't all add up to 100. But the key thing I'd like you to look at is this last column on the right, the lignin percentage. It does vary a lot based on the type of residue you're dealing with. Corn has a fair amount of lignin in it, but not nearly as much as other types of residue. Wheat has almost twice as much lignin. And so on a volume basis, on a per ton basis, wheat, for one, is harder to break down than corn stubble because it's got this more tougher material that breaks down slower than the cellulose and the hemicellulose components. Rice, in addition to having a lot of lignin, has got silica in the hulls and other places that make it more abrasive. So for years, people burned rice straw as the best way to get rid of it. Now, you can't do that in all areas, and so there are problems with decomposition. Warm season grass, not the hay, but the residue. If you let it die off or you kill it, the residue that's left has a lot of lignin in it. The stalks are very tough. And I've included newspaper. I was telling Daryl a little bit earlier, we've run into folks in different parts of the country who've been convinced that they can improve their organic matter in their soil by adding shredded newsprint. And there are folks that have actually done that. That is a source of organic matter if you get it broken down. The problem is, one, it's got a very high lignin content. Essentially, newsprint by itself has the same chemical properties as putting out sawdust or chunks of hardwood in the field. So it breaks down very slowly. It's very tough material. And so it's going to be out there a long time before it's decomposed. So that's usually a poor choice for a source of organic matter in a field situation, just because it's very difficult because of this lignin content. Corn causes trouble mainly because of its volume. There's so much residue. By itself, there are tougher residues in terms of their chemical composition, but it's the volume of corn residue that causes the problem. In terms of the decay microbes, it's the microbes in the soil that do the decomposition and break the residue down into its chemical components so we can return nutrients to the soil and form organic matter and have all the benefits associated with the decayed residue. There are three main families of microbes that are involved. Now, actually, the bacteria are a secondary decay organism. We hear a lot about bacteria when it comes to forming soil structure and converting nutrients over to forms that plants can use through nitrification. Nitrosomonas and Nitrobacter are the two key bacteria families for nutrient conversion. But in terms of decay, the bacteria really take a back seat. They're a secondary set of organisms. It's your fungal species and a higher form of bacteria known as actinomycetes that really do most of the decay work. And it's not just one type of fungus or one type of actinomycetes, but there are actually many, many different families. And then within each family, there can be several species. So we're talking about dozens and dozens and dozens of different types of microbes. It's not just one specific microbe that does the work, but there are lots and lots of different types. Some of them are actually visible to the eye if you have a compost pile in the backyard, for example, or you've got a spot in the field where you've had residue decaying in a clump and you open up that pile. Near the surface, you'll see sort of fluffy gray cotton-like groupings or colonies, and those are the fungal species at work. They'll work near the surface, and you can actually see those. The actinomycetes are fibrous in nature, and if you open up the center of the pile, they tend not to work near the surface, but more within about 4 to 6 inches from the surface. And you'll see sort of like spider webs or filaments running through the pile, and those are the actinomycetes. Anytime you see soil that's been disturbed and you get that fresh earthy smell, that's the odor or aroma coming from the actinomycetes. If you have that aroma, then you know they're present and they're active. The bacteria are invisible, they're microscopic, but anytime you open a compost pile and you feel heat generated, it's from the bacteria. There are different types of bacteria that work at different temperatures. The lower temperature activity comes from the bacteria that generate the heat. As the temperature warms, those guys die off and they're replaced by different types of bacteria that can work at higher temperatures. So there are lots and lots of different species of these microbes. They're all working together, but the most important ones are the fungal and the actinomycete species. They're also the groupings that are most disturbed by tillage. So one of the things that you'll see occur is that there's a trade-off in terms of tillage when it comes to residue decay. There's an initial bump you'll get from tillage, mixing the soil. So for example, if you run a chisel plow through corn residue, you'll typically get a faster initial decay because you're mixing the soil with the residue and exposing more of that residue to the microbes that are in the soil. That advantage doesn't last, however, because the tillage does disrupt the microbial colonies, especially the fungus. And so by disrupting them, you'll get less decay longer term through the decay process. And so typically tillage, although it provides an initial bump, is going to actually disrupt the decay process. So that's one of the advantages the no-till guys have in decaying residue once they get the system working and you've got very active soil biology in the field. So there are lots of these guys working. The actinomycetes and the fungi do most of the heavy lifting, and the bacteria are there to sort of clean up. How do they work? As soon as you have residue in the field, the combine's through, it's more or less like opening up the buffet line for the microbes. They're going to multiply very rapidly and start to work in seeking out carbon, basically as their main fuel source. Like any living body, just like us, or if you're increasing the size of a livestock herd, As you add numbers, you're adding carbon because they're carbon-based entities. There are other biochemicals required for life. We all know we need protein and all sorts of other things that basically are nitrogen-based compounds. And so in addition to the carbon, you're going to need nitrogen, phosphorus, potassium, your other micronutrients. Nitrogen is typically the limiting nutrient in addition to carbon. And we'll talk a little bit more about this later. But as the microbes multiply, and they can multiply very rapidly, a single bacterial cell within a few days can multiply to be several million. So the decay process ramps up very quickly under the right conditions. And we'll talk about the conditions here in a moment. But if there's enough moisture and enough temperature, the microbes multiply in the soil, swarm over any of the residue that's at or touching the soil surface. And so what you see in a field like this that's got corn residue that's been combined, the microbes are going to view that again as sort of a buffet. It's open season, and they're going to seek out the carbon, and they're going to use that carbon to multiply and increase their numbers, and accordingly break down that residue as they go about their work. They will need those additional nutrients, and that can be a problem, as we'll talk here shortly. The carbon that's released from the residue, 2/3 of it is basically belched out by the microbes and released to the atmosphere. It's only 1/3 of that carbon in aerobic decay that's returned back to the soil, first as part of the bodies of the microbes, and then later, as they die off, it becomes part of the soil chemistry and contributes to the organic matter in the soil. So it's only a smaller fraction of the carbon in the residue that's actually returned. Most of it's released as CO2 back into the atmosphere. But once that decay process is finished, the microbes start to die off, and then they release their nutrients, the carbon, the nitrogen, the other things, back into the soil, and then all of those nutrients are available for crop production later in the year or for the next season. So that's the process. And what we want to do is talk a little bit about this idea that the nitrogen has to come from somewhere. In addition to carbon, the microbes are going to need about 1 pound of nitrogen for 8 pounds of carbon used in this growth and reproduction process for decay. And if there's not enough nitrogen in the residue, the microbes are going to find it somewhere, and so they're going to latch on to nitrogen that's in your soil, leftover fertility from the previous year. It might be in the soil organic matter as part of the mineral profile of the soil. Wherever it's from, if they're going to keep growing and multiplying, they're going to need that much nitrogen to keep supporting their growth. And if it's not available in the residue, they're going to go and latch on to it from these other sources, and it becomes immobilized or unavailable for crop growth. And that's one of the problems that's very common, especially in corn production, because you've got a residue that's relatively short in nitrogen to begin with. And if they're tying up or locking up nitrogen from these other sources, it's not going to be available for cover crop use or for the next crop until you get that decay activity completed. To help describe that, we've used what we call the residue smile. It's this brown and green figure you see at the bottom of this slide. The brown part, the hill-shaped curve, represents the level of decay activity in the field, and the green cup-shaped curve represents the amount of available nutrient relative to the decay activity. So if you start on the left, when the decay activity is just starting, any nutrients available in your field are going to be there and not immobilized. But if the residue is short on nitrogen, and as the microbes multiply and ramp up and they're building carbon into their cell walls, they're also going to need proteins and these other biochemicals for their own life systems. They're going to take the nitrogen from the residue first, but they're also, if they're short, going to latch onto it wherever they can. And those nutrients that were previously available for crop production in the field are going to disappear because they're locked up and immobilized by the microbes. So as the decay activity ramps up, the amount of available nutrient is going to decline until you reach the peak of that process, and the maximum amount of nutrient is immobilized or unavailable. Once you get past that peak decay activity and things start to slow down, the microbes die off. They release those nutrients, including the nitrogen, back to the soil. It's then available for plant use, and that process is known as mineralization. Bottom line is if you're short on nitrogen and other nutrients in the soil, and it's typically nitrogen that's the limiting factor, you want to get this process done as soon as possible so you don't have these nutrient unavailability issues when you're trying to raise the next cover crop or the next cash crop. And so the faster you can complete the decay activity, the better off it is for your nutrient management system. Three key factors determine how fast that residue decay occurs. Carbon-nitrogen ratio is usually your best predictor for the speed or the rate of decay of any crop residue, corn residue or others. Temperature and moisture are also extremely important, and we'll talk about those in a moment. But for now, the carbon-nitrogen ratio is how much carbon I have relative to the amount of nitrogen in the residue. Almost all plant residues have roughly the same amount of carbon. It's 40%. So if you look at a corn stalk laying in the field, it's about 40% carbon. See a wheat straw laying there or soybean residue, they're all about 40% carbon. The nitrogen content varies. So some residues have more nitrogen than others. If you talk about red clover residue, so you cut the hay and instead of baling it, leave it there to decay, that has relatively high nitrogen, so it's got a carbon-nitrogen ratio of 20. For decay purposes, what we want to target is somewhere between 20 and 30 as being ideal. The maximum decay rate occurs about 25 to 1. So if your residue or what you're trying to break down has less carbon relative to nitrogen, its carbon-nitrogen ratio is less than 25, those materials are going to decay very rapidly and you're not going to have any problems. So things like red clover residue, beef manure by itself without bedding has carbon-nitrogen ratio 17 to 1. So the manure by itself is going to break down. Where you run into trouble with beef manure is if you got a lot of bedding in it, corn stalks or straw or other things that were used as bedding. That increases the amount of carbon relative to nitrogen, and that's when that manure is slower to break down. As we move up the list, we get into things like ryegrass cover. So if you spray it with glyphosate, kill it off, and leave the cover there, that's 36 to 1. It'll decay a little bit slower than the clover, but not as slowly as things like corn residue, which is 60 to 1. Small grain straw, wheat, oats, and rye tend to run between 70 and about 100. Again, the carbon content's the same. It's just that the small grain straw tends to have less nitrogen relative to corn or relative to clover. And since it's the microbes seeking out the carbon to help multiply their numbers, but also requiring nitrogen, if they run short, it's going to take them longer to do the job. Basically, the microbes themselves, if you look at the bottom of the list, are built on an 8 to 1 basis. For each 8 pounds of carbon that goes into building microbe cell walls, you need about a pound of nitrogen for the protein and the other biochemicals required for life. So if you're working with something like rye straw that's 10 times as much carbon, unless they have another source of nitrogen, they're not going to ramp up their activity and multiply very fast, and the decay is going to occur much more slowly. So that's why the carbon-nitrogen ratio is probably the most important determinant for how quickly you're going to decay residue. We talked about newsprint a little bit earlier. Newsprint's high in lignin. Its carbon-nitrogen ratio is also about 580 to 1. So you're talking about starving your microbes. If you're trying to break down newsprint, it's basically got the same carbon-nitrogen ratio as hardwood. So if you take wood chips and throw them out in the field, they're going to break down at about the same rate. There's very little nitrogen. So in order to break that material down, the nitrogen is going to have to come from somewhere else. You're either going to have to apply it or it's going to be locked up from soil sources and unavailable for crop growth. And so people that we've known that have used things like newsprint as a way to supplement their organic matter find that stuff lasts out there for years and years and years, mainly because the decay process has been slowed. And it also is going to cause nutrient tie-up problems and starve the crop while you're trying to break down those things. Sawdust by itself has a carbon nitrogen ratio of 200 to 1. So if you've ever done any work in the field and left sawdust as and just left it there as residue, you'll notice that sticks around for a while as well. Not standard practice, not recommended, but it basically comes down to how much carbon you have relative to nitrogen to determine how quickly things break down. The agronomists have worked on statistical models based on actual observed decay rates in the field, and then from that you can roughly estimate how fast you would expect residue to decay. So for the carbon nitrogen ratio, we can look at how different crops would decay based on expectations. So what I've done is based on an average temperature of about 65 degrees Fahrenheit, not too warm, not too cool, a fairly moist situation where you've got about 2 inches of water available in the soil per week. Depending on the different residue, you're going to have different decay rates. The blue line, wheat straw, has a high carbon nitrogen ratio of about 80, so it's going to be much slower. And after about 40 days in the field, you're only going to break down about 40% of it, even under ideal conditions where you've got a pretty good temperature and plenty of moisture available. Corn has less carbon relative to nitrogen, so the bacteria are going to be able to ramp up faster, work faster on the residue, break it down more quickly. And so after about 40 days under those conditions, you'd get about 60% of your corn residue. And then rye crop cover, because it's got relatively more nitrogen, you'll get about 80% of that in 40 days. So depending on the residue you're trying to break down, it really is determined by this carbon nitrogen ratio and how much nitrogen is available to support the life activity of the microbes involved in the decay process. Corn residue is present in much higher volume than wheat residue, but because of this lower carbon nitrogen ratio, it actually decays faster than wheat straw. And that's one of the reasons, for example, we were talking a little earlier about situations where you've got double crop soybeans coming in after wheat. If you don't get that straw distributed evenly and you leave a mat behind the combine, it's not unusual to come back after the beans have emerged and see variations in height and crop quality. Right in that straw mat, the beans had a tougher time emerging. There might be allelopathy effects where the residue is actually having a negative impact on the growth of the plants. Plus, you're going to have nutrient issues right in that matted area. Out away on the ends of the head where you didn't have the straw building up, usually beans are going to be taller, more robust, better color because of the nutrient availability. And so that's just one evidence of the impact that residue can have on the next crop that's growing. Wheat straw being very difficult to break down and it takes longer can cause a lot of troubles, especially in a double crop situation like that. Second factor that really is important is temperature. The warmer the temperature in general, the more decay you're going to have. Bacterial and microbial activity in the soil increases by about a factor of 2 for each 10-degree increase in temperature. Of course, if it's freezing or low temperature, you're not going to have very much decay. So if we put in a 32-degree temperature here, you'd have a flat line right across the bottom indicating that there's nothing going to decay. As you increase the temperature, you're going to have more decay. So what we've got here is a situation with moderate moist conditions for corn residue. And as we move up the line from an average 50-degree temperature where you'd expect to decay about a third of the residue in a 40-day period, if that temperature goes up to about 65 degrees, you're going to get close to 60% of it decayed in that same 40-day window. And if we had an extremely warm period like we're experiencing right now, where your average temperature might be closer to 80, It's hard to sustain that over a 40-day period, especially in the fall, but if you could, you'd get about 80% of that residue broken down in about a 6 to 7 week period. So the warmer it is, the better, until you get to about 90 or 95 degrees. And at that point, the microbes start to slow down, and if it gets really warm, they'll die off and they won't be active. So usually within the range of temperatures we're working with in the fall, warmer is better. If it gets too cool, there's not going to be much decay activity. And we always tell our people not to worry about applying our product or working to improve decay once you get daily highs below about 50 degrees. That means your average temperature is going to be getting close to maybe 40 degrees or less. And at that point, there's just not going to be much activity. So the key thing we find for trying to treat residue and speed up that decay process, the sooner it can happen after harvest, especially if you've got warmer temperatures, the more effective you're going to be. And so temperature's the second critical factor, and then the last one is moisture. Typically, the more moisture you've got, the more decay you get. Relatively dry situation where you've only got about a ½ inch of rain per week. That doesn't sound dry to us right now. We've been dry for about 6 weeks. But even at a ½ inch of rain, in 40 days, you're only going to decay about 15 to 20% of corn residue in a situation where you've got average temperatures of about 65 degrees. If you increase that to about 1.5 inches of rain available per week, you're going to get about 45% of your residue. So that decay rate more or less triples. And then if you could get a very wet situation where you had roughly 3 inches of rain available per week, average temperatures, corn residue will break down about 80% in 40 days. So more moisture, more better. If the soil top 2-3 inches is bone dry, there's not going to be any decay. The residue microbes are going to have retreated into the soil. They're going to be present in very small numbers. So the residue is going to sit there until it gets enough moisture to decay. That's why we tell people in a dry year, if you're experiencing drought right now, you probably want to wait on applying a product like ours to increase residue decay, because there simply won't be much decay. Wait till later in the fall if you get some rainfall, or wait till you've had some snowfall over winter, because the moisture is also critical. Of course, too much moisture can be a problem as well. If you start to saturate the soil, then the aerobic microbes that are involved in decay can't operate. They need oxygen. And so once the soil is saturated, you still will get decay, but it's going to be anaerobic or without oxygen microbes involved. They're much less efficient. They work slowly, they multiply slowly, and they're inefficient and tend to release more of that carbon to the atmosphere, and less of it goes into the soil where you want it. So you definitely don't want an anaerobic situation. Can't really control that. Other than trying to improve the airability and water percolation through your soil. Things like no-till farming, cover crops, anything else you can do to reduce compaction, improve air and water movement through the soil is going to help the residue decay as well. So those are other factors. About the only additional factor that has an impact is soil pH. If you've got a fairly acidic soil, microbes don't like that, and they're going to multiply and work in smaller numbers and more slowly. But if you can get close to 6.8, 7, somewhere in the neutral range, that's going to be more beneficial, and you're going to have more microbial activity, and more decay is going to happen faster. But really, the moisture, temperature, and the carbon-nitrogen ratio determine how quickly you're going to be able to decay residue in the field, whether it's corn stalks, wheat stubble, soybeans, anything else. They all basically are driven by those 3 factors. Ideally, the soil moisture content should be about 60 to 80% of its water holding capacity to get maximum decay. So it does depend on your soil as to what its water holding capacity is. Sandier soils are going to have slower residue decay because it can't hold as much moisture. Loams and clays are going to be able to hold more until they get saturated, and then you're going to drown out those microbes, and you're going to switch to anaerobic decay, which is slower and less efficient. So More moisture, more better usually, but there is a limit to that as well. Second thing we want to talk about is how much residue is out there. The old standby we used to use for years when we were dealing with lower corn yields was about 5 tons per acre, but that's good for about 150 bushels of yield. Yeah, so if you have poor soils, you're in a climate that's not as conducive to corn production, or you have weather problems that end up with a short year, you might be closer to 5. Pushing with modern hybrids, good weather, and you're getting over 200 bushels of corn, you're probably in the 10-ton range of dry matter. So that's without moisture in the residue. How much dry matter do I have? Of course, soybeans produce a lot less, and the small grains produce a lot less volume than corn. So you typically don't hear as much about soybean residue because it's there in low volume, and it's got a relatively low carbon nitrogen ratio. There's plenty of nitrogen there, and you'll be able to feed those microbes, support their life systems, and the decay of soybean residue is going to occur very quickly. Oat and wheat straw are not there in large volume, but they've got a very high carbon nitrogen ratio, and they're going to have to find that nitrogen somewhere. If it's not present, the immobilization is going to kick in. You're not only going to lock up nutrients, but it's going to take a long time to decay those residues, even though they're not present in large volume. We can get a more refined estimate, and again, we'll focus on corn residue because that's where most of the problem is, most of the concerns we hear. One very rough rule of thumb based on yield is you're going to have about 60 pounds of dry matter residue per bushel. So if you have a 200 bushel yield times 60, that gives you 12,000 pounds or 6 tons of residue. We can refine that a little bit by adjusting for test weight and moisture content. And again, we're worried about dry matter residue. The idea here is that if I take my dry matter yield for grain, how much corn do I have in terms of pounds or tons after adjusting for test weight and moisture? The 1-for-1 rule says I'm going to have just as much residue as I produced grain. So if I had a 200 bushel yield, 58 pound test weight, that tells me how many pounds of corn I had. I can divide by 1 minus my moisture percentage. So if I harvested at 16%, I'll divide by 0.84, and that tells me I had closer to 7 tons of residue. So we can refine that based on what your field conditions were at harvest. We won't talk about the small grain residues in detail, but just as a rough guideline, wheat, oats, rye produce more residue than 1-for-1. You're going to get about 30% more residue than you had dry grain yield because there's less grain relative to the amount of residue that's produced. The tillers and the stalks and the other things left over after small grain harvest. So you could basically use this 1-for-1 rule, figure out your wheat yield, multiply by the test weight, adjust for the moisture, and then increase that by 30% to get an idea about how much dry matter residue you'd have in a wheat field. Corn's 1-for-1. The small grains produce a little bit more residue relative to the grain yield. We can also refine this by adjusting for the height of the crop and the stalk quality. Usually you get about 1/2 ton to 1.5 tons per foot of corn residue per stalk height. The half end, it would be drought damaged. Corn with fairly light stalk quality would be about 1 ton per foot. And if you've got moderate stalk quality, you're talking about sort of typical numbers would be 1.2 tons per foot. And if you've got very heavy stalk quality, you're going to get about 1.4 tons per foot. So if I've got sort of typical corn, 10 foot tall, I might have as much as 12 tons. The height rule is going to give you a higher estimate, but between the 1-for-1 and the height-based rules, you get a fairly good range of numbers for how much residue you're dealing with in the field on a tonnage basis. From that, we can figure out how many pounds of nutrient I've got available in the residue, multiply the tonnage by the pounds of nitrogen, phosphorus, potassium per ton, On corn residue, we're basically dealing with about 20 pounds of nitrogen per ton and about as much potassium. Um, there is still some phosphorus. A lot of that was removed in the grain, but you'll still get about 5 pounds per ton of phosphorus. But most of the residue nutrient content is going to be nitrogen and potassium. If you know you got about 10 tons out there, you can count on having roughly 200 pounds of nitrogen, roughly 50 pounds of phosphorus, and somewhere in the 200-pound range on the potash side. That not only helps you for nutrient planning for the next crop, but it also can tell you, am I going to be short on nitrogen when it comes time for the decay process? Will I immobilize some of those nutrients, or do I have enough available? So one of the first questions we always get is, should I be adding additional nitrogen, and how much should I add? Basically, it depends on that carbon-nitrogen ratio compared with how much nitrogen I have in the residue. I'm going to tie up that nitrogen if the microbes need more than is available in the corn stalks that are out there. How much carbon enters the soil can be determined by figuring out your dry matter poundage. So DM is dry matter pounds, 12,000 pounds per acre. If you think you're going to decay half of that, the PCT value there is the percentage of the residue you think you're going to decay. So if you're going to get half, I've got 12,000 pounds out there. Half of it I'm going to decay. That gets me down to 6,000 pounds of residue. 40% of the crop residue is carbon. So if I've got 6,000 pounds, 40% of that's carbon. 2/3 is going to be lost. I'm only going to recapture 1/3 of the carbon in the soil, so then I multiply by 1/3. And then we know that once I have the carbon number, I can divide that by 8, because I'm going to need 1 pound of nitrogen for 8 pounds of carbon to multiply and reproduce those microbes and build new ones to help with decay. So that's going to determine the amount of biomass nitrogen required for the decay activity. For example, suppose I got a cornfield with 9 tons of residue at 20 pounds of nitrogen per ton. I've got about 180 pounds of N out there in my corn stalks. If I'm going to decay about half of that in the next week based on my moisture and temperature conditions, I'm going to generate about 1,200 pounds of biomass carbon entering the soil. So my 9 tons of residue, half of that's going to be decayed, so I'm going to have 4.5 tons. 40% of that is going to be carbon, and then 1/3 of that is going to go back into the soil. That's where I get the 1,200 pounds. Then I divide the 1,200 by 8 to tell me that I'm going to need about 150 pounds of nitrogen per acre to decay half of that residue. I've only got 90 available, half of the 180 that's in that residue, so I'm going to be 60 short. Roughly 60 pounds per acre is going to be immobilized or locked up until I get that decay process completed. If you add more nitrogen, depends on what you're going to do. If I'm going to put in a legume cover crop after my corn and it's going to produce its own nitrogen, the lockup may not be a problem. If I'm not going to wait and do anything until spring, in which I put most of my nitrogen on early in the spring, tying up what's available in the fall may not be an issue, because there'll be plenty of nitrogen available from your next application before you plant the next crop. Maybe I'm going to plant a grass cover crop, something, a small grain like annual rye. Maybe I do need nitrogen for that. So if I'm going to tie up 60 pounds, and that's really going to limit the availability for the annual rye crop in the fall, I might have to put on some in order to help the decay process, make up for this mobilization, and make sure that I've got enough available for my cover crop. So whether you add additional nitrogen or not depends on how much is going to be locked up and whether you really need that immobilized nitrogen or not for the next crop, whether it's a cover crop or something you're going to do in the spring. Hey, maybe I'm going to raise beans in the spring. I don't need that much nitrogen, and so the immobilization may not really be a factor. Second question we get a lot is, what about adding sugar? There are a number of companies out promoting applications of table sugar or molasses, other sugar sources to help with decay. The sugar contains carbon, And so it's readily available. It breaks down in the soil very quickly. The carbon is immediately available almost to help support increased microbial life, and it will help break down residue because it's providing that carbon fuel for the microbes. But there's no nitrogen in sugar. So basically what you're doing is increasing your carbon nitrogen ratio out in the field, making it a tougher job for those microbes to break down if there's not enough nitrogen available. So you're either going to have a bigger lockup problem, more immobilization, or you're going to have to add nitrogen to balance out the sugar. From our experience, you're better off to avoid that problem and not increase your carbon nitrogen ratio, but try to speed up that residue decay process in other ways. So sugar can help, but in another sense, it can also make the job bigger and slow down the decay process if you don't have enough nitrogen to support the microbial regrowth that sugar is going to try to promote. BT corn, we get this all the time. The guys here at No Till Farmer had sent out an article a couple of weeks ago about BT corn. Almost everybody agrees that BT corn stalks are harder. It contains almost twice as much lignin. The agronomists generally all agree on that. Does it take longer to decay? You talk to a lot of farmers and they believe so. Lignin is more difficult to break down, so there is a good argument to be made that it would take longer to break down, but also the carbon nitrogen ratio's not really much different from BT to non-BT corn. So looking at carbon nitrogen relationships, it should take about the same time. And the study that Daryl and John and the guys here at No Till Farmer distributed showed that there really wasn't much difference between the decay rate of BT and non-BT corn. There are several other studies done in the last 10 to 15 years that report the same thing. Interestingly, there are almost as many studies that show that BT corn does take longer. And the most recent articles by some of the more careful researchers point out that there are a lot of other factors that are probably influencing these results, whether you see a difference or not. The lignin content is not really a big difference. It's probably driven by something else. Some cases, BT corn has smaller residue quantities than non-BT corn. You're going to get more stalk and more height in particular out of non-BT varieties. There could be differences in insect infestations and stalk quality based on insect feeding. All sorts of other things that can't be controlled from one experiment to another. Moisture content, temperature, the initial level of biological activity in the field or the lab wherever this research has been done. And so the bottom line is it's really hard to say whether BT corn takes longer to decay, and most of the evidence points to no, it doesn't take longer to decay. It is harder, but it basically has the same carbon nitrogen relationship, and it probably takes about as long. Most of the variation you see is probably due to these other factors like temperature and moisture and biological activity. Very quickly, we did want to talk about our approach to the problem. We market a product called Chandler BioCat 1000. Like all of the other products we market for Chandler, they're enzyme based. Ours is unique. There are other there are other biological treatments for residue on the market, but most of them use live bacteria and other cultures in the treatment. Ours is enzyme based, which means it works on all of those microbes, the fungus, the actinomycetes, the bacteria. It's designed to increase their reproduction and their activity, so you get a broader spectrum increase in your microbial activity in the field. The product includes enzymes and amino acids, all of those nitrogen-based things that are required to help support the life functions of the microbes, so you have fewer problems with tie-up. It doesn't contain some of those other micronutrients that are required. They're chelated, so they're available. And the product also has a natural surfactant to help it spread across the residue and get down to the soil layers. So even if you're not incorporating your residue in a no-till situation, it's going to work into the soil surface and get those microbes going where they're starting from, the top 2 or 3 inches of the soil. By multiplying those microbes and getting them working faster, you can get through that immobilization phase, complete the decay process faster, and start to release more of those nutrients for the next crop, whether it's a cover crop or a next cash crop in the spring. We've had very good results over the past several years, as long as you're in a situation where you've got adequate moisture, adequate temperature, and there's not something else in the field that's going to be inhibiting biological life, pH problems, stuff like that. But as long as you've got moisture, temperature, and the enzymes can help the microbes do their work, you can decay residue pretty quickly. This picture in the upper right shows fairly typical results. This is residue taken from a treated field. The product was applied in the fall. Next spring we went back. The untreated residue on the left is more or less as it was left in the fall. We've got cross sections of the stalks there showing that most of the pith, which is largely soft cellulose, is still intact. The stalks on the right, most of the pith is decayed and gone. The stalks are hollowed out, and the outer shell, which is largely composed of the lignin, is still there to provide soil cover to prevent wind and water erosion. But the stalks are hollowed out, they're brittle. You hit them with a tire, you hit them with a planter opener, they're more or less going to shatter and break and not cause any problems with planting operations or tear up your tires, things like that. If you've got dropped ears in the field, it's going to decay as well from the inside out. It's going to take care of the center of those cobs first, the softest material, work to the outer part of the stalk where you've got, or the cob where you've got more lignin, but eventually it'll get out to the germ of the kernels. The cobs will be there, but they won't germinate, so you'll have fewer problems with volunteer corn and things like that. We recommend applying about 16 ounces per acre of BioCat 1000 as soon as you can get it on after harvest in the fall. Put it on by itself. It'll mix with our soil product, Chandler Soil, and it's also tank mix compatible with most liquid fertilizers like 28% nitrogen. If you're putting on burn down chemicals, either fall or spring, it'll mix with that, and just make sure you use enough water to get good coverage. And at our current discount price, it's somewhere between $7 and $10 per acre, depending on the volume that you're purchasing at and also the rate. Like we did in our first webinar, we'd like to extend a discount offer to the participants. Normally, our best price of the year is what we're offering right now. Through the 1st and the 15th of September, we have a 12% discount. If you're watching the webinar, you've participated, we'll gladly extend that discount to you through October 31st. So if you call us up or you send in an order and we know you were in the webinar, we'll extend that discount through the end of October. We offer the same discount from December through the first week of January. So if you want to think about it a little bit, usually you'll be able to get the same price in December as well. But if you're buying for the fall, but you want to do so later, we'll give you the 12% off through the end of October. Other than that, we really appreciate your time and listening to us. We see some questions here in the room, and other than that, Daryl?
No, thank you very much, Doug. A lot of good information there on residue decomposition, the process, what's all involved in it. Obviously, it's a big thing for no-tillers, so it's a very informational session here you provided for us. Why don't we do have a few minutes here. Why don't we go ahead and look at some of the questions that are up here. I think what I'll do is we'll just run through. I'll let you go through and pick out some to answer, and if we run out of time, we'll get the rest of them, to you so you can provide some written feedback here later. So, but no, go ahead, and maybe what you want to do is just read back the ones that as you answer them. So.
Okay. Well, the first question we saw up there, Dan wanted to know about breakdown on leaves. Corn leaves have relatively less cellulose and higher lignin content because there's less pith in the leaf and more structure. Leaves tend to break down fairly quickly because of their smaller volume. But in terms of their lignin content, they're tougher material. But again, the carbon nitrogen ratio is about the same as it is for the rest of the plant. So leaves tend to break down fairly quickly, and usually what you're left with at the end is the root crown and the base of the stalk to provide cover for the soil. A lot of the stalk tops, the leaves, break down early, and then the cobs go next. So leaves actually contain a lot of lignin, but they break down quickly. Mark is asking about lignin content in wood chips depletes soil nitrogen when added to the soil during decomposition and the breakdown process, and that's right. The lignin's not only tough, but it's also the very low nitrogen content of wood chips. The microbes won't be able to ramp up and work on wood chips very quickly because they're going to basically not have any nitrogen available. So they're going to have to take it away from the soil. They're going to deplete that soil nitrogen while they're trying to break down things like wood chips in the soil, and that's why it takes so long. Very high carbon nitrogen ratio. It will seek out, the microbes will seek out soil nitrogen where they get it. So there's going to be a high amount of immobilization anytime you're trying to break down any sort of wood-based material in the soil. After corn harvest, Mark wants to know about do microbes get a boost from added exposure to light, air movement, and moisture conditions? Once you get rid of the main part of the residue and broken it down, pushed it over with a combine, you will have more airflow. Moisture, it can be a plus or a minus depending on evaporation rates. What's really critical is how air and moisture move through the soil, not necessarily through the field. So if you've got a soil that's relatively open, drains well, you're going to have better decay. If you've got a compaction problem where air and water don't move as well, the residue is going to break down more slowly because it's going to be harder for the microbes to get going. Crop roots, what's the carbon nitrogen ratio and what sort of factor would you estimating root mass? About the only number I've seen there came out of the University of Missouri several years ago. And on corn, the root mass represents about 40% of the total residue left over after the corn crop. So you've got almost as much below ground as you've got above ground. It varies by crop. Grass type crops like corn are going to have more because you've got a more extensive root system. Things like native grasses will be even higher than corn because the roots extend more deeply and it's a more extensive root system. Something like soybeans, you may have a large tap root, but it doesn't extend out as far, and so the percentage on soybeans is going to be smaller. A larger share of the residue is above ground. Let's see, Mark wants to know about carbon residue, providing soil cover and conserving soil moisture. That's right. That's one of the reasons you don't want to burn up all of the moisture or all of the decay and decay all of the residue right away in the fall. You do want soil cover over winter, not only to help prevent erosion, but also to help manage moisture content. And as well, that's why you want organic matter in the soil because it's one key source of water holding capacity. And so if you break down the residue too fast, you're actually going to provide less erosion protection and have problems with moisture management down the road.
We'll do one more question, Doug, here. Go ahead.
Question is about why we don't recommend sugar. Basically, we've had very good results without using sugar. And the key thing is that when you add sugar, you are providing an energy source for the microbes, but you're also increasing that carbon nitrogen ratio because you're not adding nitrogen with the sugar. There's plenty of carbon in sugar, but no nitrogen. So sugar by itself actually increases that carbon nitrogen ratio. It is readily available, but unless there's another nitrogen source available, it's going to start to lock up or immobilize the nitrogen available in the soil, and it actually hampers that decay process. Um, so the sugar by itself helps speed things up initially, but it actually makes the process last longer than it would. Um, the decay process is it's natural. It's gonna take time, even under ideal conditions. I think a lot of farmers are looking for a product that's going to go out and more or less melt the residue, like putting water on um, cornstarch peanuts or on pouring it on popcorn or something like that. You just want it to melt and go away, but you really don't want that. You want some soil cover. You don't want the residue to decay too quickly, or else you're going to have problems with your organic matter in the soil. Um, you want to leave that outer shell of the stalk as protection, but you do want to break it down on the inside to make sure it's brittle, release as many of those nutrients as you can before you need them, and make sure they're available for the next crop, and avoid problems with things like punctured tires and clogging up row openers on a no-till planter and things like that. So we really want to manage the rate of decay to make sure that it's going along at a good pace so we can avoid immobilization, but we don't want to decay everything because we do need that soil cover.
Real good. Thank you, Doug. Thank you. Excellent. Appreciate you going through this topic with us today. We are going to conclude at this point, and I just want to give you a couple heads up that when you log off today, you're going to receive a questionnaire is going to pop up to complete, and it's really simple, very easy to complete, a small number of questions. If you don't mind answering those, that's going to help us determine the value of today's webinar and also help us with planning for future webinars. Second, we will be making available via email to you a replay of today's webinar in the event you'd like to go back and review the entire presentation or even some parts of the presentation. We'll also be putting that replay up on our No-Till Farmer website shortly. And then again, any questions, and there were a few that we didn't get to. We got to most of them, but there are a few we didn't answer yet. We're going to share those back with Doug to answer, and we'll post those along with the webinar replay. So again, on behalf of Doug Miller and also today's producer, Cole Vandermouse, sure want to thank you for joining us today and for following No-Till Farmer. And hey, we're heading into harvest season here, so I hope you guys have a real safe and a great harvest, and have a great rest of the week. And we'll look forward to seeing you on another webinar shortly. Take care.